All right, let's finally get into the sponges. So we're going to cover the phylum Porifera now. And as you can see from this outline, it's the usual sort of objective. So we're going to talk about the brief background to the Porifera first, a little bit about their taxonomy and phylogeny. We're going to go into the general characteristics of all sponges. And after we talk a little bit about each of them, then we're going to go into each of the different poriferan groups and talk about what features make each of them unique and easily identifiable from the other groups and key adaptations. So first, if we get into the taxonomy and phylogeny of the sponges, remember now we're into the metazoa, well into them, and remember that the definition of the metazoa is that they're multicellular, motile, heterotrophic eukaryotes. And I bring this up again because when you look at sponges, it's often hard to really imagine that these things are really animals. They don't seem like animals at all. They seem like slightly sort of living rocks or something. But in fact, they really are living organisms and they are animals. So they're multicellular, they're motile, at least at some point in their life. They're definitely heterotrophic and they're clearly eukaryotes. Okay, this is a phylogeny you saw earlier when we talked about the protozoans. Here's some of the amoebozoa. This whole clade here is the Opisthokonta. And we didn't talk about most of these, but we're already well into here, into the metazoa. And if we zoom in, here's the phylogeny for the Porifera. Notice it's composed of 3 different classes, the Calcarea, the Demospongiae, and the Hexactinellida. Now, all sponges are characterized as having a series of pores and canals that allow water to flow through them. And in fact, the name Porifera derives from the Latin for pore, so having many pores or many holes through them. Each of the classes is diagnosed based on the composition of their spicule. So the spicules are these structures that form what can be loosely referred to as the skeleton of sponges. It is sort of an endoskeleton in the sense that it's internal and not external. But it's basically these rigid structures that we'll go over in more detail in just a moment. So we'll come back to the phylogeny in a moment, but it's important to note that the Demospongiae and the Hexactinellida are each other's closest relatives. So they're sister taxa, and sister to them together is the Calcarea. Okay, so first let's go into the general background of sponges. So clearly they're multicellular. So I say here, what's so great about that? Well, by virtue of being multicellular, multicellularity allows you to grow bigger and to grow much bigger. If you remember those single-celled protozoans, the problem with being single-celled is that if you wanted to get bigger, either to compete, to get more resources, to defend yourself, a single cell can only get so big. And the reason is because the bigger it gets, the harder it is for nutrients or key elements to go through the membrane and get to where it needs to be inside the cell. So there are limits to how big a single cell can get. So one way that you can overcome that is to become multicellular. So you have many cells that actually come together and then they begin to work together, function together, and they can basically perform tasks for each other. That's going to lead ultimately to the formation of tissues, which by definition have to be multicellular. And then tissues will begin to organize into a division of labor such that you then see organs developing and then organ systems. And we're going to see this as we go throughout the semester. So being multicellular is a huge innovation. As I indicated, it leads to what's referred to as incipient tissues. So what that really means is it's the precursor to being true tissue. So you have different cell types, so you're multicellular, you have different cells that are doing different things, but they're not necessarily all working together universally for each other. So it's the beginnings of the tissues world. So what do you know about sponges? There are a lot of different species of sponges. They come in a lot of different colors and shapes, as I'm going to show you here in a few different pictures. There's quite a bit of diversity in the sponge world in terms of their texture, their composition, their colors, and where you even find them. So currently there's approximately 5,000 known species and more being described all the time. So they're all aquatic, primarily marine, but some are freshwater, found at all depths. We generally think of sponges as being sessile, and they are sessile. They're usually sitting at the bottom of the ocean floor or the freshwater floor. But those are usually the adults. The embryos have a tendency to be free swimming, and you can find them moving about in the water column. As you saw from those pictures, their general shape kind of depends on the substrate that they're living on and the vagaries of the water currents where they're living. They do exhibit a number of different symbiotic relationships. So you find them engaged in commensal relationships and parasitic relationships with other organisms. So many animals live among the sponges commensally. And sometimes there are parasites that live on sponges. So they feed off the sponges without the sponge gaining anything. In fact, if anything, it loses out on that situation. There aren't a whole lot of enemies of sponges, but there are some organisms that do eat sponges. And certainly we harvest some of them for our own uses economically. So they actually are important to us economically. Although these days most sponges are commercially produced synthetically, so we're rarely actually using real live sponges anymore. But when we did, back in the day, we used them, and they were all pretty much all in the class Demospongiae. Okay, let's talk about the general organization of sponges. So again, they are literally a system of pores and canals. They have water flowing through them, and that water flows through them, and they actually build up the current to make that happen. And that's to bring water through them to bring food and to help them to expel their wastes. So there's three general what we call shapes of sponges. You can be asconoid, cycanoid, or leuconoid, and that's in an increasing order of complexity. So this is a diagram from your book showing the asconoid shape. So this is the simplest shape. And what you're seeing is if you do a cross section through the sponge here, so this is the outside of the sponge, this is the inside, what's happening is you have a system of pores that allow water to enter and then canals that enter into the sponge. And then the water exits through the middle, through the osculum. So you're going to enter through this, the porocyte cells allow you to go in. The hole is referred to as an ostium. And then this is going to be referred to as the spongiceal, and water will exit the osculum. And so in the asconoids canals or shape, it's a very simple system of water going in through the sides and out through the center. So the water flow I'm showing you here is through the into the ostium, through the porocyte, into the spongiceal, and out through the osculum. We're going to talk about some of these different cell types that you're seeing here in just a moment and tell you what they do. But just to give you a sort of punchline, these cells right here are called choanocytes, and these little flagella are actually beating to make a water current. So the flow is actually drawing water into and then out through the osculum by virtue of those flagella moving. So the spongiceal is lined with these choanocytes. I just said that. And I just said that. Okay. The second shape is the cycanoid shape. So, fascinating. I guess I had those slides in there twice. Okay, here we go. Cycanoid. Okay, so what's happening here is this is a slightly more complicated shape compared to the asconoid. And you can almost think of it as if someone had taken the outside of the asconoid and if they had folded it in and out and in and out, almost like you would do a fan when you were a kid, that's what would make this type of shape. So what they're showing you here is a cross section through the side of the sponge, and it's blown up here. And let's go through the water flow. And there's a number of different terms that you need to know here. So you start out here, and you're going to actually go in here to this in-current canal. You're going to enter into this sort of outer sort of canal of the cycanoid sponge by going through this hole right here that's referred to as a prosopile. Here it is spelled out. So water goes in the prosopile into this radial canal. If it goes through this hole called the apopile into the spongiceal, and then it goes up and exits through the osculum. So into the ostium, in-current canal, prosopile, radial canal, apopile, spongiceal, and out the osculum. So we have all these terms, and we often create a lot of these extra terms in biology because we're trying to be really specific in communicating with other scientists. That's why we have these different terms. So also, it's important to note these choanocytes, and we're going to talk about the choanocytes here and how they actually not only set up the water current, but they're bringing in food particles, and they're the ones that are going to take in the food particles to allow the sponge to feed. But it's important to note that when you had an asconoid, those choanocytes were only in the spongiceal in the center of the sponge. Here, they're in the radial canals, and that has a consequence that we'll talk about in a minute. Here's the last form. So this is leuconoid, and this is the most complex form. What you see here is basically take that last form, the cycanoid, and double it up and kind of squish it in, and you've got it. So water goes in the incurrent canal. Through the ostium first, into the incurrent canal, and then it goes into this complex system of flagellated chambers here, all lined by choanocytes. And ultimately, the water will flow into these excurrent canals and out the osculum. So you no longer have that central sort of tower-like spongiceal. Now you have a much more complicated sponge with these flagellated chambers instead. So just to give you a heads up, the reason that this is important is because what's happening is by virtue of losing the spongiceal and then developing these flagellated chambers, you're increasing the surface area with which you can feed. So what that means is that the sponge can get bigger and bigger and bigger with these flagellated chambers. When you had the simple sort of asconoid shape, you can only get so big and you can only take in so much food. So this increasing complexity also allows you to get bigger and feed more efficiently as well. Okay, so there's a number of different cell types that you're seeing now with the sponges that you didn't see with the cnidarians nor with the protozoans. So what you're seeing now is increasing complexity. So you're developing different kinds of cells. Different sorts of cells are doing different sorts of functions, performing functions for the bodies of these sponges. And so this is ultimately going to lead to tissues, but you don't really have tissues yet because the cells aren't necessarily all working together uniformly. So what you see here in this diagram is here's a close-up on these choanocytes. So these are flagellated cells that are going to serve to, as the flagella beats, it's actually going to set up a water current. It's also going to draw food particles into it, and I'll show you how the choanocytes feed. The outside of the a sponge body is covered by these pinacocytes, and so they don't have much ability to move or react. Remember, these sponges don't really have a true nervous system yet, but they can actually allow pores to sort of open and close to allow water to come in and out. They have some ability to do that. Archaeocytes, I'll explain in a bit more detail. These actually can perform all kinds of functions and transform themselves. They're almost like the stem cells of the sponge world. So these guys can actually they're sort of amoeba-like in their movements, and they do quite a bit of feeding and digestion as well. Colonocytes are actually cells that are going to provide the basis for collagen deposition for certain kinds of spicules. And then you're actually seeing some spicules here. These are sort of generally drawn as these little sort of needle-like structures. They can be composed of either collagen, spongin, calcium carbonate, or silica. And then the rest of the body of the sponge, in between the outer layers and the inner layers, this is all composed of this sort of mesoglea called mesohyle. It's this sort of gelatinous matrix. So let's go through each of these cell types in a bit more detail. So here's a choanocyte. This is the nucleus and sort of the body of the choanocyte. This is the collar, and here's the flagellum. So what's happening is the flagella is kind of whipping around, and all of them are beating sort of in unison. And by virtue of that whipping motion, it's creating this water current that flows through the sponge. When that happens, food particles are drawn in. So sponges are feeding on all kinds of small food particles that are in the water column. They could be a variety of bacteria, plankton, broken down detritus, all kinds of stuff that's floating around in the water column. You can actually see what's meant to be food items here. So these little food particles here on the sides of the collar. And what happens is they get trapped. So this collar actually functions like a sieve. It's drawing water through and into it, which makes the particles get trapped. But the water continues to flow through, so the current continues going. And then there are these microfibrils here in the collar. And there's this action of drawing the food particles down the collar and into the body. And so what happens is food vacuoles then pick up the food particles here in the body of the choanocyte, draws it into the choanocyte, and ultimately will draw it into the body of the sponge, where it will be broken down intracellularly. These pink cells are archaeocytes. And so as I indicated before, they're sort of amoeboid. They sort of move like an amoeba through, they're not an amoeba, but they move like an amoeba through the inside of the sponge. They move through the mesohyl. So they phagocytize particles. So the food particles go in through the body, these choanocytes, and then into the mesohyl. These archaeocytes then grab them, and then they break them down. Now, these archaeocytes can also differentiate into many different other types of cells. So they can become sclerocytes, where they form spicules. They can be spongocytes, which produce spongin, which is a sort of flexible connective sort of tissue, but it's not a true tissue. Or can form cholinocytes, so collagen. So basically, they can form different kinds of spicules, is what it really amounts to. The pinacocytes you can see here in sort of purple. So these are the cells of the external epithelium, although it's not a true epithelium because it's not a true tissue, but it's the precursor to epithelium. So it's sort of like the outer skin of a sponge. It's somewhat contractile, so it helps regulate the surface area somewhat, but more importantly, it helps regulate the flow of water through the sponge. And you actually see them forming into these pores and porocytes. So the porocytes, slightly different. So pinacocytes would be here on the edges. The porocytes would be here. Okay, and that's these cells right here. So these are tubular cells and asconoid sponges that allow water to flow into the sponge. So I showed you these sort of general stylized spicules here. These, again, there are many of these inside the mesoglea that sort of gives the sponge a bit of rigid structure so that it can grow up from the bottom of from whatever surface they've actually landed on. They can be composed of three different things. They can be composed of calcium carbonate, of silica, and then of spongin or collagen. And so you find that these are defining features for the three classes in part of the sponges. We're going to come back to classification in just a minute. Let's talk in general about how they get it done. And I'm going to show you some really interesting videos in class, but I'm not going to show you them here in this lecture. We'll see them in class. So in terms of digestion, we already talked about that. So the food particles get trapped on the collar. They get transported down into the body's food vacuoles, then take them into the body of the sponge, and then the archaeocytes phagocytize them. Oh, just to reiterate, food particles can be detritus, which is just broken down materials from something that's died. It can be plankton, either phyto or zooplankton, or bacteria. Okay, what about their excretory system? So these guys don't have a full excretory system like we have. So what they do is they use vacuoles to take those broken down waste products and then release them out through the pinacocytes. So it's diffusion via contractile vacuoles. In terms of locomotion, well, so most of these guys are sessile as adults, and they don't really move much. The larvae are modal, and I'll show you a bit of that in just a minute. Some species are known to sort of slightly crawl. It's not very much, but they can crawl. But it's very slight movement. They don't have a nervous system at all. You're not really going to see the beginnings of a nervous system until we get to the cnidarians in the next lecture. Again, in terms of respiration and osmoregulation, so dealing with carrying of oxygen and water, this is again carried about through diffusion via contractile vacuoles. And they don't have a circulatory system. So even though these guys are a bit more advanced than the protozoans, they're still quite simplified organisms. It's almost hard to believe that they're animals. So what about reproduction? We haven't covered that yet. So there's a couple of new terms, new concepts I want to introduce you to. The terms are monaceous and dioecious. So in many animals, you can have both sexes on a single individual. So you have male and female parts, and those are referred to as being monaceous. You have both on the same individual, okay? Or you can be what's called dioecious. So you have separate sexes. So humans are a great example of being dioecious. So you have males and you have females. Now, a term that often is synonymous with monaceous is hermaphrodites. So basically, being hermaphroditic is the same thing as being monaceous. So you have male and female parts on the same individual. However, what you'll find throughout the animal world and also in the plant world as well is that if you are monaceous, you aren't necessarily able to have functional male and female parts at the same time. Sometimes you do, but usually either those parts are not actually active at the same time. So maybe you have pollen, say on a plant, you have pollen that goes out, but maybe your female parts aren't receptive at that time, or maybe the female parts are receptive, but you're not making pollen anymore. So it's the same with animals. So often you're what's referred to as being either a sequential hermaphrodite, meaning that you have both parts, but you're not using them at the same time. You use one first and then the other. Or you can be facultative, so you can be you can be sequential or simultaneous. So simultaneous would mean that you would have both parts and they're functional at the same time. Okay, well in the sponge world, most sponges are monaceous, so they have both male and female parts on the same individual, but there are some that have separate sexes. And I'm going to show you some interesting videos about reproduction in sponges that are really cool in class. But just to give you the bullet points, so some sponges, what they'll do is they'll expel their sperm into the water column. And then because those flagella are and those choanocytes are beating and creating that water current, that current draws the sperm into the female or into the other sponges that have eggs. The sperm will then fertilize the eggs in the sponges, and then they'll actually release the fertilized eggs into the water column to hatch and then form these free-swimming larvae. So in some cases, it's the male's releasing the sperm. Usually, sponges cannot do what's called selfing. They can't fertilize their own eggs. Some can, most cannot. And in some cases, there are other species where they'll release eggs and sperm at the same time into the water column, and then they'll unite and then float up through the water column. So in this case, another term you need to be familiar with is oviparous. So these are, whoops. Oh, it looks like part of the slide got cut off. So when you're oviparous, you reproduce using eggs and you release eggs. Okay, so ovi means egg, So if you're oviparous, you're an egg layer. And so sponges are egg layers. So this is just a diagram from your book showing a free-swimming larva. It's a parenchyma larval form. And ultimately what will happen is after they've grown to a certain stage, they'll then settle out onto the surface of the lake or stream or ocean. They'll begin to divide and grow until they eventually, in this case, form a type of demospongia. You can see what's meant to be these flagellated chambers here. So what's important to note is that sponges can reproduce both sexually and asexually. And I say jokingly here, but when the going gets tough, the tough mate without mates. Actually, for most animals, if you can reproduce both asexually and sexually, generally what happens is you will reproduce asexually when times are actually good, and you'll try to get your numbers way up. And get your numbers up so that it's easy to find a mate. And then when times are bad, that's when you might actually try to mate sexually. And when times are bad, that's usually defined as being that the environment's gone wrong for you somehow. So maybe things are drying up or there's not a lot of food around. That's when you have a tendency to mate sexually, because then by genetic recombination, you have much more genetic variability in your offspring. And then they are usually more genetic diversity means you're better able to adapt to changing environments. When things are good, you don't worry about that so much, so you can reproduce asexually. It doesn't matter if there's a bunch of clones around. Okay, so I say here that all sponges are capable of reproducing sexually and asexually. What I didn't mention, though, is that there's an interesting way that sponges can reproduce asexually. Generally, when they do it by budding. So we talked about what budding was in the protozoans. But they produce these structures inside their bodies called gemmules. And these gemmules are basically little embryos that have a hardened sort of outer surface. And so they're almost like a little cyst. And they can withstand really harsh environmental change. So they can stay sort of buried in the muck for a really long period of time until the environment is better and more suitable for growth. And at that point, they can begin dividing and developing into a free-swimming larval form, which will continue to feed until it's ready to settle down on the surface of the environment that it's in. Okay, let's talk about the classification of sponges. So here's that tree again. You should commit this tree to memory. So you should know that the Hexactinellida and the Demospongiae are sister taxa, and sister to them are the Calcarea. So what you'll notice from here is that the first key characteristic of each of the, so now we've covered the general characteristics of sponges. We're now going to talk about the unique features of each of the three classes. So the class Calcarea is characterized as having calcium carbonate spicules. So it's very scratchy feeling if you were to touch it. It can be any shape. It can be asconoid, cyclonoid, or leuconoid. It can be any of the three. So just because you're seeing what looks like a simple sort of asconoid shape here doesn't mean that that necessarily is true for all the members of the Calcarea. The Demospongiae make up about 80% of sponge species, and they're showing you a diagram here, but they can take on a number of different shapes, although primarily they're all leuconoid. They can be different other shapes, but they're primarily leuconoids. That's the most complicated shape. The spicules can either be made of silica, of spongin, or both. So what that means is that your sponge is going to be a little bit squishy if it's got spongin, but if it's got some silica spicules in there, it's going to be kind of scratchy. So I've known people to pick up sponges on the beach and say, oh, look, and then they scratch it on themselves, and it's like they've just slit themselves open with little pieces of silica. The spicules in this case do not have 6 rays, and I'll show you a 6-rayed one in just a moment. These are mostly marine. And here's an example of a demosponge in the ocean. The Hexactinellida are really interesting sponges. So these guys are incredibly deep sea, and you find them off of the coast of Australia. Very deep sea sponges. They have spicules made of silica with 6 rays. And so these are often referred to as the glass sponges, and they're extremely fragile. They're very, very brittle, and they can break easily. We only have one in lab, so please guard it with your life. So Calcarea and demosponges, they tend to be rather asymmetric in their symmetry, whereas most Hexactinellida tend to be radially symmetric, at least in part, in their upper bodies. They can exhibit either cyclonoid or leuconoid body shapes. And remember, so both of these two can have silica in their spicules, but the Hexactinellida have 6 rays. The demosponges do not have 6 rays. And I guess I lied, I don't have a picture of this, but there is a picture in your book of what the 6 rays look like. So this is the tree that I showed you before, the roadmap for the class. So we've covered the protozoans and the porphyrians, and I think earlier I referred to the cnidarians as having already been gone over, but clearly we haven't covered that yet. In theory, you ought to be able to get this very tree, and you should be able to fill in the blanks in these trees. And what I'm indicating here is like the clade name that's in this sort of dotted box, so you should be able to tell me what the names are, and then you should be able to tell me what major features diagnose or are characteristic of the clades above it. And so here we are with the porphyrians. So as we go through the semester, see if you can try and fill in these blanks as we go along. And I may only have one listed, but often there are more than one characteristics that could be in those boxes. Up next will be the cnidarians. All right, that's it for the sponges. Thanks for watching. And if there's any questions that I haven't answered, please always be sure to ask me, fill out those worksheets, and let me know how it's going.